All right, 17.2, evolution as genetic change in population. Let me get rid of this. So I love this one. Knowing the lion's preference for red meat, the spamalopes remain calm but weary. Spamalopes. Do you ever try spam? Ugh. Alrighty. Let's go on here. Our goal is to discuss mechanisms of evolutionary change other than natural selection, such as genetic drift and gene flow. Our objectives are to explain how natural selection affects single gene and polygenic traits, describe genetic drift, and explain how different factors affect genetic equilibrium. So, the insect populations often contain a few individuals that are resistant to a particular pesticide. Those insects pass on their resistance to their offspring, and soon the pesticide-resistant offspring dominate the population. This relationship between natural selection and genetics explains how pesticide resistance develops. So our first objective is to explain how natural selection affects single gene and polygenic traits. So evolutionary fitness, a couple of vocab words for you, is the success of passing genes on to the next generation. Evolutionary adaptation is any genetically controlled trait that increases an individual's ability to pass on its alleles. So natural selection for a single trait can lead to changes in allele frequencies and then to evolution. For example, a mutation in one gene that determines body color in lizards can affect their lifespan. So if the normal color for lizards is brown, a mutation may produce red and black forms. If red lizards are more visible to predators, they might be less likely to survive and reproduce. Therefore, the allele for red coloring might not become common. Single gene traits, for example, their allele for red, single gene traits, why did I say that twice? Anyhow. Black lizards might be able to absorb sunlight. The higher body temperature may allow the lizards to move faster, escape predators, and reproduce. So the red, obviously, you can see it. The other animals can eat it very easily, and then so the black blends in better, so it's going to reproduce. And that definitely makes sense, doesn't it? Polygenic traits have a range of phenotypes that often form a bell curve, which we've seen before. Skin color and height, for example. The fitness of an individual may vary from one end of the curve to the other. Natural selection can affect the range of phenotypes and hence the shape of the bell curve. And this next two things are important when it comes to polygenic traits. Directional selection, you need to know all these, occurs when individuals at one end of the curve have a higher fitness than individuals in the middle or the other end. The range of phenotypes shifts because some individuals are more successful at surviving and reproducing than others. For example, if the only large seeds were available because there was some drought or something like that, then like on the Galapagos, the birds with the larger beaks would have an easier time feeding and would be more successful in surviving and passing on their genes. So the whole graph shifts, all right? Directional selection. Stabilizing selection occurs when individuals near the center of the curve have a higher fitness than individuals at either end. This situation keeps the center of the curve at its current position, but it narrows the overall graph. So you have less extremes. You have more in the middle, less extremes. For example, very small and very large babies are less likely to survive than average-sized individuals. The fitness of these smaller or larger babies is therefore lower than that of more average-sized individuals. Now, this is the last one, disruptive selection. This occurs when individuals at the upper and lower ends of the curve have higher fitness than individuals near the middle. Disruptive selection acts against individuals of an intermediate type and can create, eventually, two distinct phenotypes. For example, in an area where medium-sized seeds are less common, birds with unusually small or the birds with unusually large beaks would have higher fitness. Therefore, the population might split into two groups, one with smaller beaks and one with larger beaks. You can think about what might happen with that. So you need to know the three types of selection when it comes to polygenic traits. So our objective one, explain how natural selection affects single gene and polygenic traits. Natural selection on single gene traits, it's quite obvious. They can lead to changes in allele frequency and thus to changes in the phenotype frequencies. Natural selection of polygenic traits, a little more complicated, can affect the distribution of phenotypes in 3 ways. Directional selection shifts the whole graph over. Stabilizing selection just gets more broad in the middle and less on the ends. Or disruptive selections, where you have less in the middle and you may end up with 2 distinct different types of populations then. All right. Objective 2, describe genetic drift. Genetic drift occurs in small populations when an allele becomes more or less common simply by chance. Genetic drift is a random change in the allele frequency. Now, there are some ways that this can happen. The first is the bottleneck effect. The bottleneck effect is a change in allele frequency following a dramatic reduction in the size of a population. You can think about what things might cause a dramatic reduction. Drought, terrible, like a tsunami or something. A disaster may kill many individuals in a population, and the surviving population's gene pool may contain different gene frequencies from that of the original gene pool. So then you would end up with differences, all right? And the next, this is the bottleneck. I should have clicked on, sorry. So this is an example they give. You have the parent population. Only some of them come out. Only some of them survive this devastation. So these surviving individuals and they're left with their genes reproduce, and you end up with maybe more of one of the other. And the founder effect is the next thing. The founder effect occurs when allele frequencies change as a result of the migration of a small subgroup of population. That's what the word means, founder. Like some left and they became the founders of a new population. 2 groups from a large diverse population could produce new populations that differ from the original ones. So let's say you have a founder here and you have a founder here, and then the descendants all look like that and all look like that. Maybe they live in 2 different areas and 2 different environments too. So describe genetic drift. In small populations, individuals that carry a particular allele may leave more descendants than other individuals just by chance. So over time, a series of chance occurrences can cause allele to become more or less common in a population. That is genetic drift. So objective 3, explain how different factors affect genetic equilibrium. Oh yes, the Hardy-Weinberg principle. So a population is in genetic equilibrium if allele frequencies in the population remain the same. If allele frequencies don't change, the population does not evolve. The Hardy-Weinberg principle describes the conditions under which evolution does not occur, which means the population will not evolve. Let's go through this. The principle states that allele frequency in population will remain constant unless one or more factors cause those frequencies to change. The factors are large population. Genetic drift can cause changes in allele frequencies in small populations. Genetic drift has a less effect on large populations, such as the ants shown. Large populations help maintain genetic equilibrium. Next, no mutations. If mutations occur, new alleles may be introduced into the gene pool and allele frequencies will change. Next, random mating. All members of the population must have an equal opportunity to produce offspring. Individuals must mate with other members of the population at random. In natural populations, I'm not going to say anything. However, mating is not random. For example, the female peacocks choose mates on the basis of the physical characteristics, such as the brightly feathered tails. Such non-random mating means that alleles for those traits are under selective pressure. That doesn't happen naturally. There's going to, random mating. Not gonna, go for it. No. Anyhow, here's another thing that has to happen. No movement into or out of the population. Individuals who join a population may introduce new alleles to the gene pool, obviously. Individuals who leave may also remove alleles from the gene pool. Thus, for no alleles to flow into or out of the gene pool, there must be no movement of individuals into or out of the population. Next, no natural selection. All genotypes in the population must have equal probabilities of surviving and reproducing. No phenotype can have selective advantage over another. Meiosis and fertilization do not change the relative frequency of alleles in the population. The shuffling of genes during sexual reproduction produces many different gene combinations, but does not alter the relative frequencies of alleles in a population. So there you go. Those are the Hardy-Weinberg principles, the 5 conditions. Again, according to Hardy-Weinberg principle, the 5 conditions that are required to maintain genetic equilibrium is the population must be very large. So larger the population, the less change. 2, there be no mutations. 3, there must be random mating. 4, there be no movement into or out of the population. And 5, no natural selection. So our objectives are explain how natural selection affects single gene and polygenetic traits, describe genetic drift, and explain how different factors affect genetic equilibrium. Lots of information in this lecture. So all of them support our goal of discussing mechanisms of evolutionary change other than natural selection, genetic drift, and gene flow. Alrighty, guys, let's go over here, our pretty little picture of some bugs this time. Enjoy.